This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Hello everybody, good afternoon, and welcome to our second part on our Module 1 single cell analysis. Today, once again, we are very lucky to have an invited lecturer, and a really awesome one at that. And I'd like to introduce to you Dr. Davis McCarthy. Dr. Davis McCarthy is head of the Bioinformatics and Cellular Genomics Lab at St. Vincent's Institute of Medical Research. And his main research is focused on developing new machine learning and statistical methods to high throughput process biological data, as well as applying them in different scenarios. And he has contributed to several different methods, and very popular ones, on differential RNA-seq analysis, as well as single-cell analysis. So, Davis, I know how busy you are. Thank you so much for accepting this invitation to talk to us.
Thanks, Douglas. It's a pleasure to be here. Always happy to talk about single-cell RNA-seq analysis. As Douglas said, I'm taking over from Pete to give the second half of this week's material on single-cell analysis. I have less facial hair than Pete, but I am a little bit taller, so swings and roundabouts and all that. So from what I understand, Pete took you on the first steps through an analysis garden in single-cell RNA-seq data, and gave you an introduction to some of the key steps to start with, so going from the FastQ data, the raw sequencing data that we get out of experiments in this area, through sort of the big picture of what we want to do and can do with some of these exciting single-cell technologies that have been available now for the last sort of 5 or so years and are really gaining in popularity. Pete took you through some of the important steps like read alignment and quantification, talked through quality control and normalization with some of those packages up there. So of course I developed the SCADA package, so it's gratifying to see that's still being used by many people around the place. And today I'll take over where Pete left off, which means starting around this point in our tour through the analysis garden. So I'll start by talking about dimension reduction, how we use that, why it's useful in single-cell RNA-seq analysis and some of the specific approaches that we take. And then I'll talk about a handful of downstream analyses. In the limited time that we have, I'll just touch on 3 sort of important topics that we want to cover in downstream analysis, namely clustering, trajectory inference, and differential expression. And so when we get to those parts of the talk, I'll explain all of those sorts of things in more detail. So to start with, let's talk about dimensionality reduction. So this is obviously not unique to single-cell RNA-seq analysis. Dimensionality reduction techniques are extremely useful in just about every area of data analysis that you can think of. They're particularly useful in genetics and genomics because what we generally get are high-dimensional data sets, meaning we have hundreds, thousands, tens of thousands of measurements made on each individual sample, which in our setting here, we typically think of that as a single cell. And so here, we typically have, after QC, perhaps 10,000 to 15,000 gene expression values present for each cell that we've assayed. And so we can't look at that data and make sense of it. Can't open Excel and eyeball that data to get a feel for what's going on. So if we want to understand the large-scale structure in one of these high-dimensional data sets, a really useful thing to do is find an appropriate means to reduce the dimensionality of the data set into something that we can visualize and then perhaps interrogate in more easier fashion. So some popular methods, generally speaking, for dimensionality reduction are MDS, so multidimensional scaling, and PCA, so they're very classical statistical approaches to dimensionality reduction. You almost certainly have encountered them before. PCA, principal components analysis, is like sort of the canonical approach to dimensionality reduction. And just to refresh you, if you have not thought about it in a little while, what we want to do with PCA is find linear combinations of the variables that we've measured that essentially define new axes through the high dimensional space that capture in the first principal component as much variation in the data as possible. And then when we compute the second principal component, we want to do the same thing, find another axis of variation by a linear combination of variables with the constraint that it is orthogonal to the first principal component. And we can keep doing that to get further and further principal components. So PCA is linear, because we're taking a linear combination of the variables. That has many strengths. It's deterministic, meaning if we repeat the PCA calculation on the same data set, we'll always get the same answer. It's sort of easy to understand. It explicitly orders the principal components by the amount of variation in the data that I explained. That's a useful property. But for some of the data sets we look at, especially in biology, not all the effects that we might want to capture and view in our low-dimensional space will be linear. So in recent years, some nonlinear approaches have become popular. And so TSNE, I'll explain what that is in a minute, has become a really popular tool in the single-cell field, as well as elsewhere in kind of machine learning and statistics, as has UMAP, a uniform manifold approximation projection approach. And both of those methods have become really popular because they're able to capture some nonlinear effects which can be useful in many settings. So the picture shown here is a TSNE plot. So TSNE stands for T-distributed stochastic neighbour embedding. I don't have time to even unpick that particular name, let alone give you a detailed idea of what it does. But the very brief intuition behind TSNE is that it tries to model essentially the distances between data points in the high dimensional space as conditional probabilities, and it tries to spread out the points in the low dimensional space, say in two dimensions, by again using the sort of idea of conditional probability, in this case with the t-distribution, to spread out the points in this low dimensional space. For this particular data set, we've got a set of blood cells from Dr. Christine Beiben at WeHi, just across the road. And so you can see that the TSNE plot in this particular case does a really nice job of separating the different cell types. And so in this particular case, we have annotations from the biologists based on, say, marker genes, using what they know about their biology, where we can have sort of truth labels about what's going on, and we can see that these particular cell types cluster very nicely in this TSNE space. And this is a particular feature of TSNE as a dimensionality reduction method. When it works well, it has a tendency to cluster points together. And so when we have data sets, which is quite common in single-cell RNA-seq, where we have many different distinct cell types present, TSNE often creates very attractive representations of those data sets because here the clustering that is immediately visible in the 2 dimensions here of TSNE maps very directly to the underlying biology in this case. We can look at the same data set with MDS. So multidimensional scaling, another very classical statistical approach to dimensionality reduction. MDS has many sort of associations with PCA, which I described before. MDS is a bit more general in the sense that you can define a distance between all the pairs of points in your data and give that distance matrix to MDS, and it will find the right representation of your cells so that the low dimensional representation reflects the distance matrix that you give it. So it's more flexible than PCA in the sense that you can define distance in many different ways and give that distance matrix to MDS and it will give you a plot back. So when we look at this data set, we can see that sort of this approach in this case and what we've used Euclidean distance in this case, which makes this essentially equivalent to PCA. Compared to PCA on this particular data set, we separate the granulosomes here well from the erythrocytes here, but other cell types are sort of all clumped together here. So just in the first 2 dimensions of MDS here, we don't capture all of that biological structure that seems obvious when we've got these distinct cell types. But what we can do with MDS, of course, is to look at later dimensions as well. So if we then look at dimension 3 and dimension 4, those sort of purple, orange and blue cells that were clumped together in the first 2 dimensions here are nicely separated in dimension 3 and 4. So whereas TSNE could separate all these cell types quite easily in 2 dimensions, in the MDS plot here we need to go to a few later dimensions in order to separate the different cell types that we know to be present in this particular case. So again, various approaches, all very useful for taking, in this case, hundreds of cells on which we've measured 10 or 15,000 genes and their expression level, so a very essentially large, high dimensional data set, and we can compress a lot of the information into 2 or 3 or 4 dimensions and actually really get a visualization that helps us understand the large scale structure of the data set. To talk a little bit more about TSNE, because it's ubiquitous in the single-cell RNA-seq field now. For a time it felt like perhaps you were legally obliged to put a TSNE plot in every single-cell RNA-seq publication. They were that common. It works nicely when you have a large number of cells. So if you have 50 cells, TSNE typically won't give you a particularly appealing plot. There'll be a tendency maybe just to sort of have a ball of points. But when you have a large number of cells, and also when you have diverse cell populations or cell types in that large number of cells, then it can do a very nice job of grouping points in what is often a very appealing way. So this is another version, another data set, also blood cells, so PBMCs, about 3,000, almost 3,000 here, produced from the 10x Genomics platform. And if you've encountered PBMCs before, then you'll see familiar cell types here. So we have a couple of different types of T cells present, natural killer cells, dendritic cells, megakaryocytes, monocytes here, different type of monocytes over here, and the B cells distinctly over here. So again, in this one plot, we get a big overview of the data set. And, you know, if you're analysing single-cell RNA-seq data set and you're collaborating with biologists and you give them a picture like this, they'll generally ooh and ah, and they'll be very pleased and think you're super smart and doing a great job. So that's handy. But there are a few things to be aware of when doing TSNE, and especially when talking through these sorts of results with collaborators or other people. And one of the main issues with TSNE, apart from it being stochastic in the sense that if you run the algorithm again on the same data set, you'll get a slightly different picture. Often that can be just a trivial difference, like flipping the axes or something like that. Sometimes it can be more profound. But the bigger issue probably with TSNE is that it can be very easy to overinterpret results for less heterogeneous data, or even for heterogeneous data like this. And we've got some results here. So what's quite nice about TSNE, it was created by Lawrence van der Marten and Jeff Hinton. If you've not yet spent time in the machine learning field, Jeff Hinton is a very famous figure in machine learning. They're quite honest about what it is. They say TSNE is a way to create nice pictures of high-dimensional data sets, and it can do that very well in many settings. And as they note in this particular sort of blog/article, which is very well worth your time, although TSNE plots are often impressive, they can be very tempting and easy to misread. And the main things to point out there are that there are a few hyperparameters involved in producing a TSNE plot, and the values of those hyperparameters matter a lot. So what we're showing in these pictures are, so this is a little example data set from this publication. So this is the original data set that they then transformed in various ways and then given back to TSNE to produce a plot. And so there's a parameter in TSNE called perplexity, which is related to Shannon information, but you can think about it, roughly speaking, as kind of the number of nearest neighbours you expect each point to have. So a low value of perplexity, you expect points sort of not to have too many close neighbours, a high value of perplexity, and points will have many close neighbours. So perplexity of 2, you get this quite sort of spread out thing where the points in the different groups are not closely connected to each other. As you increase the perplexity, you sort of get much, much tighter clustering. So you can see if you were to arbitrarily choose a perplexity of 2 or perplexity of 30, you would get quite different impressions of what the structure of this data set is. So that's an important thing to be aware of. Similarly, unrelatedly, cluster sizes and locations in a TSNE plot don't have any inherent meaning. So PCA is really nice to interpret because it's kind of linear and it's honest about the distances that it presents. That's not the case for TSNE. TSNE just tries to spread out essentially the distinct points it finds as much as possible to kind of use as much of the lower dimensional space as it can to show you the structure of the data. So that means that it kind of shows local structure but not global structure in a TSNE plot. And so this example shows that. So here we have 3 distinct clusters in the original data. 2 of them, the blue and the yellow, are quite similar to each other, and the green's quite different. And so if we try with different perplexities, so perplexity of 2 doesn't work at all well for this. Perplexity of 5 starts to spread out the distinct groups a bit better. Perplexity of 30 groups them very nicely, but they all look kind of equally distant from each other in this particular plot, and that's a feature of TSNE. So it will tell you that all of these blue points are similar to each other, all of these yellow points are similar to each other, but we've got to be really careful about interpreting sort of how far or how different yellow is from green, green is from blue and yellow, etc. So that's an important point for interpreting TSNE. We can't trust the distances in the low dimensional plot that it shows us. So cluster sizes don't mean anything, whether they're tight or spread out. You've got to be careful not to overinterpret. Distances do not mean anything. Random noise doesn't always look random, so you've got to be really careful with that. Often you can see shapes sometimes, and again, the tendency to overinterpret is always there. If we have a sort of complicated topology for a data set, we may need more than one plot to understand it, and particularly we may need to look beyond TSNE to do that. So TSNE everywhere in single cell data analysis, so a good one to be aware of, and it has its ability to create really attractive plots, especially when there's a lot of diversity of cell types in a data set, but also comes with some risks. But forget about TSNE, no one uses that anymore. It's passé. The new thing's UMAP, which is a uniform manifold approximate projection, or something like that. Has many characteristics of TSNE, except it's faster, and it is actually sort of a true dimensionality reduction technique, unlike TSNE. I won't explain that, but if that piques your interest, I'll let you go and look that up. UMAP often creates pictures much like TSNE. Like TSNE, it has a tendency to group points together by cell type. This is pancreatic data in this particular case. So again, we have very distinct cell types present, and they group themselves together in a nice, attractive way in this particular UMAP plot. So I won't go into this one any further, but my group at SVI, joint with Melbourne Institute of Genomics, a couple of weeks ago presented a single-cell RNA-seq workshop over 2 days. All of the materials are publicly available online, so we have a latent spaces chapter in that workshop, so if you want to read a little bit more about TSNE, UMAP, and some of these other techniques, along with some code to play around with it, then the link's there, so go and check that out at your leisure. So we've just talked about dimensionality reduction techniques, extremely useful, come with some caveats. Like a lot of these things, when we're trying to understand how these things work, it can be useful to have some sort of reference data sets, or benchmarking data sets, so we can evaluate the performance of different methods. And So Matt Ritchie's group at WeHi, Matt gave this week of lectures last year. He and his group and many colleagues produced this paper and R package and data set called CellBench, which provides several reference single-cell RNA-seq data sets that are carefully thought out so that we have some data sets where there is some known ground truth that we can use to evaluate different single-cell RNA-seq methods. So it's incredibly valuable for that reason. And in one particular data set they did, this is a mixture data set, they extracted bulk RNA from 3 different cell lines. Don't need to worry too much about precisely what they are. We'll denote them with different colours here. And they produced different mixtures of these 3 cell lines, so from 100% H22B, 100% H1975, these colours don't quite match there, but so be it, and 100% the other one, and then varying different sort of fractions or concentrations of the different cell line RNAs in these down to one that's like equal parts, all 3 of them. So if we look at this particular point here, that's 60%, 66% green, 16% red, 16% blue. What they also did was produce sort of starting material for these mixtures with different amounts of starting RNA. So from 30 picograms, which is quite a lot, that would be a very big cell that gave you 30 picograms of RNA, down to 3.75 picograms of RNA. And so this is a really nice data set because we know what the mixtures are for different samples, we know what the amount of starting material is, and so we can think about plotting these and looking at different, what I'll show you in the next plot, different normalization methods, and this gives us, this reference data set gives us a means to evaluate their performance. So what we're looking at here is principal components analysis after normalizing the raw count data by different methods. So CPM is counts per million, TMM is a bulk RNA-seq scaling factor normalization method. SCRAN is a similar approach to TMM but designed explicitly for single-cell data, and BASICS is a hierarchical Bayesian model that also tries to do normalization of the data. And so what we're looking at here is the output of that reference data set. So again, we've got our mixtures here, and the size of the points here indicates this amount of starting material. So perhaps let's start with the one that's easiest to understand. And so SCRAN performs very well in this particular case, where the PCA, the first 2 principal components, very neatly recapitulates what we know to be the underlying sources of variation in this case. So the sort of 3 points here, we've got the pure populations of those 3 cell lines. In the middle, we've got the equal mixtures of those cell lines, and in between there, we've got those mixtures that favour one or the other. And so that's kind of exactly what we're expecting to see. What we also nicely see in the SCRAN picture is that the first principal component or second principal component are not picking out the amount of starting material, which is sort of a technical feature that a good normalization method would get at the sort of underlying biology, which is the different cell lines here, rather than reflecting the amount of starting material. And SCRAN here does a good job of doing that. That contrasts particularly to PC1 if we do CPM. So CPM, counts per million, this is kind of the simplest normalization method you could apply, where for each cell you add up the total read counts that were observed for that cell and divide all the values by that total and multiply by 1 million. So you get the counts per million. And that's an important starting point from raw counts because the amount of sequencing that we do or can do per cell varies widely between individual cells. Pete might have mentioned this on Wednesday. So it's very common for the amount of total counts or total reads observed for the different cells in your data set to vary by 1 or 2 orders of magnitude. So you might have some that have 1,500, 2,000, 5,000 counts, up to some that might have 100,000, 200,000 counts. And so if you just use those raw counts, then the main source of variation between the cells will be just the amount of sequencing that you're able to do per cell, which in general is not something interesting. So we want normalization methods that get rid of that effect, and CPM gets part of the way, but we can see that PC1 is really strongly picking up this climb from low starting material to much higher starting material. And then PC2 kind of starts to separate the cell lines somewhat. TMM is like SCRAN, so building on CPM, the idea with TMM is that instead of just dividing each cell by the total counts observed for the cell, you find a scaling factor that does a better job of the normalization that takes into account some of the other issues that arise. I won't describe them in detail now just for time, but I'll leave you to explore that at your will. So in this case, at least this does a better job than CPM in the sense that now PC1, so the main axis of variation, is differences between the cell lines. But PC2 is absolutely still driven by the amount of starting material. And we see also, basics has a similar problem in this case. So with lots of normalization methods out there, a reference data set like this is really good to try and get a handle on which normalization methods are performing well. And as you said, SCRAN is the clear winner on this particular data that extends this idea of scaling factors a bit further precisely for the single cell setting and in this particular case, and generally speaking as well, it does a good job in the normalization. So that's kind of means for a lot of the workflows we and others use SCRAN's our default approach to normalizing the single cell RNA-seq data before we go on to do further things down the line. So two really important approaches, things we need to do in single cell RNA-seq analysis, clustering and trajectory inference. So these two are related but also distinct from each other. In clustering analysis, well, both of them, I guess, in both of the cases we're trying to define the relationships between cells based on, again, some underlying biology. In clustering analysis, we're typically thinking about having discrete cell types or state present in our data set, and we want to distinguish them from each other. So like I was showing with the TSNE plots earlier, we might want to take a sample of PBMCs and we want to define a cluster so that we can define the different blood cell types that are present. For trajectory inference, we want to do sort of similar things in sort of defining cell states, perhaps, but we're thinking about them in a more continuous way. So this will arise in, say, development, say embryonic development, where we've got, in some sense, a continuous change from, say, a stem cell state to a differentiated cell type, and there's less of an idea there of distinct clusters or distinct cell types on that trajectory. There's, in some sense, a continuous change from one state to another. In that case, the continuous idea of change is what we capture with trajectory inference. So to start with clustering, there are at least 60 different methods available now for sort of single cell data, from very classic clustering approaches like k-means on principal components to kind of very specific models that people have developed for single cell RNA-seq data. With that many models, there's obviously way too many to run on every data set to try and figure out which one we like. So what's really helpful are benchmarking efforts like and reference data sets like CellBench that can provide us with some guidance on which methods to use and when. So we'll just point you to these two papers, Gio et al. And Freitag et al., both of which I would say are useful without being absolutely definitive. And I think that's fair enough because there's not going to be a definitive answer about which clustering method is best. That's kind of not exactly the right question to ask. No clustering method will be absolutely definitively the best for every situation we can think of. But papers like this can give us some guidance about probably which methods are never useful, never recommended for use, and sort of getting us into a space where, okay, here are, say, 8 to 10 methods that generally perform well and have different strengths and weaknesses for particular problems that we might look at. These are some of the methods available out there at the moment. The key ones I'll point out are CellRanger. CellRanger is a piece of software produced by 10x Genomics, which is the producer of the major platform, currently most popular platform for doing single-cell RNA-seq analysis. So the CellRanger software pipeline is kind of a default that many people will run. It has a clustering approach, so that's present there. SC3, produced by colleagues at the Sanger, is an interesting approach in that it does several different types of spectral decomposition of the data matrix, clusters them, and then looks for the consensus clustering across those different methods. So the idea there is that we get a little bit more robustness than if we were just to choose one approach versus another. It tends to work well for smaller data sets. So if we have, say, less than about 3,000 cells, SC3 tends to work quite well. It doesn't scale to very, very large data sets. Another important one to know about is Seurat. So Seurat is an R package from Rahul Satij's lab at New York Genome Center, and it's become a very, very popular platform for doing single-cell RNA-seq analysis. They use a graph-based clustering method. Don't have time to discuss the details now, but the very short description is has an approach to building like a KNN graph, a nearest neighbor graph, and then does formally Louvain, uses the Louvain algorithm for community detection to find clusters on that graph. And overall, again, in many settings, and particularly for large data sets, those sort of graph-based approaches do well. So this is just a small selection of the 60 or more tools that exist. How can we decide what works? Well, one approach is to, again, find a data set with some ground truth. This is a data set from Kang et al., published last year, a paper called Demuxlet. The nice thing here is that the idea is that we've got cell lines, again, the cell lines which have from different human individuals, so different genetic, natural genetic variation between these cell lines. We can mix these cells together, and then we can identify which cells belong to which of these cell lines based on the genetic variants, the SNPs, that occur naturally in the RNA-seq reads. So in this case, we can define, well, we can figure out which cells came from which individual based on their genetics, and we can use that sort of ground truth to assess clustering methods where we expect the different methods to cluster the different cell lines separately. So in the Freitag et al. Paper, they did this analysis and computed the adjusted RAND index. I'll let you look that up. But adjusted RAND index is a very popular way to define the similarity of two clusterings. So adjusted RAND index of 1 means the clusters capture exactly the same information, and the minimum is 0. So here we see quite a variety of performance. 3 individual cell lines, so 3 true clusters. So many methods get close to that. Some do very well on this particular data set. Some find a few too many, but are kind of in the right ballpark. And then some find way too many clusters. So race ID 2 here finds almost 80 clusters on a data set where there were 3, we think of as 3 ground truth ones. So that's useful to know that, if you're thinking about using race ID 2, it will have a tendency to find many, many more clusters than might be truly present in your data. On the other hand, this kind of group of methods do pretty well in this particular data set and kind of might be in the bucket where we're like, oh, they're all kind of plausible candidates. We can do another one, another sort of approach, 10x data here, PBMCs. So we can take sort of biological expert annotations for these cells, these different 3 data sets. So reasonable idea of ground truth. Here you can immediately see that all of the methods have much, much lower adjusted RAND index values, essentially telling us that this is a much harder clustering problem than that first one of the 3 separate cell types. And in this case, the performance metrics look quite different. So race ID 2, which looked terrible in the first data set, actually has better accuracy than most of the other methods in this particular comparison, but it still has the characteristic that it produces heaps and heaps of clusters. So here there's something like 12 true clusters, actual kind of biological cell types, and race ID 2 kind of splits those clusters into heaps and heaps of clusters, so up around like 150 or so. So again, sort of there's some information here, but also with the great variation between cell types, even sort of thoughtful comparisons can make it hard to interpret what's going on. So to dig in one of these data sets a bit further, so this is PBMCs, 10x PBMCs, looking at Cell Ranger, Seurat, Similar, and T-Scan for the methods that flashed up on the screen earlier. And these are the clusterings that they give for this data, shown in the same TS&E space. And if we zoom in on just a couple of portion of this plot, we can sort of, check how these clusterings agree with what it looks like to us based on TS&E, which again we have to take with a grain of salt. You can see Cell Ranger decides there are 2 clusters here, which kind of perhaps matches our intuition just looking at it. Seurat agrees with that particular cluster, but then splits this off into a couple. Similar mainly decides that there are 2 clusters there, but actually there's a bit of something different here and a few odd cells from different clusters scattered around. And then T-Scan decides this is one cluster, but breaks this one into 2. So at some level, if we were to say try to merge these ourselves, we could probably decide, oh yeah, T-Scan is actually kind of just split this cluster into 2, so that kind of is not that much of a disagreement. But on the other hand, there is a large amount of discrepancy between just applying these different algorithms to exactly the same data. So that's something to be aware of. For one of these combinations, we get some advice. So again, thinking about the actual biological problem that we want to solve is really important. Are we interested in rare cell populations? If yes, then race ID and SC3 can be appropriate choices because they find really small clusters, so they'll have a tendency to find potentially way too many clusters, but they might be able to split off that one rare cell population from the rest. On the other hand, if we're not interested in rare cell populations, then these graph-based clusterings like Seurat and Cell Ranger, those sort of approaches can perform very well. If you want more, and particularly like an explanation of what's going on with these graph-based clusterings in some detail, Pushui Chow from my group has put together a really nice description and summary of these graph-based clustering methods. And so again, that's available in our single-cell RNA-seq workshop materials. So the links there take you straight to that chapter on clustering and cell annotation. So that was the discrete case. In case like early development, which is we've got here. So here we've got mouse embryonic cells going from zygote to early, so the 2-cell stage, 4-cell, 8-cell, 16-cell, up to the blastocyst stage, so very early in development. And here there's much more of an idea of sort of continuous change as the zygote develops into the blastocyst. And so we could think about, so this is these cells in PCA space, and we could think about sort of charting a course through this cell population that shows this sort of continuous change. And so Ruchian, again from my group, has put together some really nice material on this, so check that out in our workshop materials. And in this case, we've applied a method called Slingshot, which again generally works quite well. And what it does is generate a quote-unquote pseudo-time value for each cell, which tries to find that continuous trajectory from the first cell state to the end cell state. And again, in this particular case, it does quite a nice job of picking out the zygote and early 2-cell cells early in pseudo-time and pretty well ordering these different cell types based on their expression in the correct order of their development. And here in this particular data set, we've got the actual labels of the different cell stages, which allows us to kind of check what we're doing. So it's, again, a nice little reference data set. Details there, as I've said. Like the clustering problem, there are probably at least 50 trajectory inference methods now. So again, that problem of benchmarking and finding a reference is really important. This paper is outstanding, one of the best benchmarking papers I've seen. They do a fantastic job of really rigorously and thoughtfully comparing all of the different trajectory inference methods across lots of different axes of things that might be important. And so if you're interested in trajectory inference, then this is absolutely the paper to read to try and figure out what might be a good choice for your particular problem. Beyond that, if you're interested in doing benchmarking of methods, then this is a fantastic paper to read because this really kind of sets the bar at the high level for for all aspects. So doing a really comprehensive survey of a field, very rigorous comparisons, and then wrapping all of that up into a completely reproducible framework that means that all of the stuff they've written is in Docker containers with wrappers and so on, so you can actually go and reproduce all of the results that they present in the paper. It's a really impressive piece of work, so a great example of what you can do. The final aspect of analysis that I'll talk about before we finish is this problem of differential expression. So I think you will have encountered this before, but the idea is that we have one or two or more samples, and we would like to compare or find which genes are differentially expressed. So have a significant or measurable change in expression between the conditions, and so we need to identify them with statistical means. So this has been a well-studied problem from microarray data to bulk RNA-seq data, and now it's also, of course, relevant to single-cell data. Methods for bulk RNA-seq data are still alive and well, so there's the EDGAR package and method, which was developed at WeHi, so that was led by Gordon Smythe. I worked on that several years ago, many years ago now, before I went and did my PhD. Lima-Voom is another very well-established approach, as is DE-seq2. So, as we'll see in a minute, these bulk methods were perhaps initially thought not to be relevant or appropriate for single-cell RNA-seq data, but actually that seems not to be the case. And then, as well as those sort of classic methods from bulk data, many methods have now been developed kind of specifically for single-cell RNA-seq data. The main one I mentioned there is one called MAST. There's still a lot sort of unsolved problems, perhaps, in questions about what's working for differential expression. What's interesting and one point I want you to take away for thinking about this for single-cell RNA-seq data is you have both designed comparisons, so where you say, here are my different experimental conditions that I know about before I do the experiment. So that's like a classical statistical setup, and most of these methods, especially for the bulk, have been designed with that sort of statistical problem in mind. You know a priori what your different groups are, and you compare those different groups with a statistical test. That's subtly different from another common problem in single-cell RNA-seq data, where we do a clustering, and then we want to say, what are the marker genes that distinguish, or most strongly distinguish, this cluster from that cluster from this other cluster? And now in that case, that's not a designed experiment anymore. You've learnt those clusters from the data. So then you have to be very careful with interpreting a statistical test, because you're basically breaking the assumptions of these statistical tests. If you've defined clusters from your gene expression data, then by definition, those clusters must have differences in expression between them, or else your clustering method wouldn't have found them. So you're biasing your statistical test. So when we're looking for those marker genes, we have to bear that in mind and basically take any p-values that come out of these tests for marker genes with a big pinch of salt, and not rely too much on them in what we do. Going back to that point of benchmarking, Sonnerson and Robinson from last year produced another very good benchmarking paper that compared a lot of these methods. And interestingly, one of the key takeaways was that they found that these well-established bulk RNA-seq analysis methods do not generally perform worse than those developed specifically for single-cell RNA-seq data. And I think we can interpret their results even more strongly to say, actually, several of the best performing methods are bulk RNA-seq analysis approaches, and in fact, only one or two of the methods developed specifically for single-cell RNA-seq data actually perform well. And so that's a bit of indictment on the single-cell RNA-seq methods development, that a lot of things were published probably without appropriate comparisons to existing methods or rigorous enough simulation or real data analysis testing. So actually it's some of the classics in the field that still work best. There are still many open questions. Of course, since I wrote EDGAR and it performed well in that comparison, I like to use it myself for my analyses, and we'll make that recommendation here. EDGAR uses a negative binomial model underneath the hood and a generalized linear model framework. What we found works particularly well for, what they found in that benchmarking paper works well, is a quasi-likelihood approach in EDGAR. If you've done any generalized linear modeling, you know that you have a mean function, and you link the mean to a linear predictor through a link function. And the other key part of generalized linear model is the variance function that sort of defines the relationship between the variance of your observed values and the mean and other parameters in your model. The interesting thing about the negative binomial model is that its variance function is quadratic in the mean, so it captures a lot of variability in raw count data. The quasi-likelihood method adds an extra parameter to that variance function, so that third parameter can model more of the variation, so it can handle even more variation than the standard negative binomial model, and that proves to be useful in the single cell setting where there's even more over-dispersion or variance in the count data than we observe, say, for bulk RNA-seq data. So our recommendation is to use the quasi-likelihood approach in EDGAR, and what they also found in that paper, that including the cellular detection rate, or the proportion of genes expressed in each cell as a covariate in the model, is also useful for increasing the accuracy a little bit. The final thing I'll touch on now is a nice piece of software for interactively exploring a single cell data set. So we talked in the dimensionality reduction at the start about how useful and important it is to visualize your data, and have some methods to do that. This package, produced by Aaron Lund and Charlotte Sonnerson and some other colleagues in Europe, allows us to explore a data set very interactively. So I'll just show you very briefly what this looks like. Of course, that would be right. It's working perfectly when I hooked up the laptop, but no. RStudios quit on me. Very well. I won't show you that. Always the risk of trying to do a live demo is that it fails on you, so so be it. But the nice thing is, so this is an R package again. It plugs perfectly into the pipeline that we've already used up to this point. So you can have an object in your R session that has PCA, UMAP, whatever, sort of information contained in the model, as well as the gene expression values and cell annotations and gene annotations. And then this will, you can run IC, it pops up a tab in your browser, and then you can look at sort of panels like this where you can say, look at PCA space. You've got visual parameters that will let you, say, color the points by whatever metadata information is interesting, like the sample or the cell type or so on. You can look at plots of the distributions of gene expression, gene expression values or other cell metadata, like the number of genes expressed per cell and things like that. What I would have shown you if the little demo had worked is that you can actually select subsets of cells. So, for example, you could, say, drag and create like a little box to say, okay, I want to know a bit more about this particular population of cells from the PCA plot, define that, and then link that set of cells in other panels. So you can say, okay, well, what's the gene expression for a particular gene just in these selection of cells? So some really nice features to kind of get your hands dirty. And again, from the position of like a collaborative bioinformatician where you might be working with biologists or other people who've generated data or set up a study, this is a very nice tool to have available to it, because it's something you can give people where they can explore their own data set, which is always really fulfilling, and also a way to talk through an analysis with some collaborators. So that's it. That's our tour of the analysis garden. Pete started you off again with the raw data. That's kind of some of the platforms in the history of where we came from leading up to the single-cell RNA-seq analysis, kind of where we are now with the platforms. He took you through read alignment, quantification, quality control, pointed out some of the tools that are available to us that we use commonly to do these sorts of analyses. Today we talked a bit about normalization, quite a bit about dimensionality reduction, and I introduced you to three particular aspects, important aspects of downstream analysis in single-cell RNA-seq data, mainly clustering, trajectory inference, and differential expression, and we talked about some of those packages that are available there. So in summary, single-cell RNA-seq is a powerful technique to study gene regulation and behaviour cell by cell. The counts that we obtain straight off the protocols that we can apply contain high levels of noise and variation, with a lot of a term that I don't particularly like, but commonly called dropouts, meaning that a gene is expressed in a cell but we miss it for whatever reason. That adds to the noise of the variation we observe. As Pete took you through, quality control is extremely important so that we remove problematic cells that have failed in processing for one reason or another, and also to get rid of low abundance genes which essentially have no information that's of use to us. What I talked about today, so dimension reduction approaches allow visualization of the large-scale structure of these high-dimensional data sets. Clustering and trajectory inference are common tasks that we want to do, and there are many tools available out there for that now. Many of the best methods for differential expression analysis for single-cell RNA-seq data are actually those that worked well for bulk RNA-seq data. And sort of a constant theme for what I've talked about today is that careful and thorough benchmarking is really essential to guide choices about which analysis methods to use when we're navigating a very large and messy analysis garden. Having good evidence-based ways to choose one method over another is really valuable for people whose focus is on doing the analyses to try and gain some insight about biological questions. Pete and I, Matt Ritchie, many others are big fans of bioconductor ecosystem, and over the last few years there's developed a really nice R-based ecosystem through bioconductor for single-cell RNA-seq data that incorporates early processing methods like SC pipe, great data classes to organise analyses, and then some of these downstream approaches as well. So with that, I'd like to thank all the people, Matt, of course, for producing the bulk of this material last year, all the others who contributed to it. I'll leave you with some further reading and say thanks for listening, and if this sort of stuff really excites you, then this is right in the area of research that I do, developing methods and applying them in collaboration with people to biological questions. So feel free to get in touch at any point if that's of interest. Thanks.
Thank you. Do you have any questions?
No. All right.
Not actually a question, but what do you think would be considering this is a cohort for the Masters of Bioinformatics, so maybe quite a few, or at least a few people here will be developing methods in the future? What do you think are the keys to success in developing new methods in bioinformatics and single cell?
Yeah, I think figuring out where there's a need is always a good starting point. With 60 clustering methods currently available, I'd think carefully before developing a new clustering method, just because that's a well-populated space where there are solutions that work kind of as well as you'd hope. So I think I like the idea of being sort of problem forward rather than solution backward. The best bioinformatics methods that I've seen have come where people have been faced with a challenge where no good solution exists, and so they build one. Things that don't work so well, which I've also seen, are sort of, maybe statisticians or machine learning people who have a particular model that they like and then kind of try to force that to solve a problem that may or may not exist. So I think that problem forward approach, like figuring out where there's a need for something, where there's an unsolved problem, that's the best space to be working in. And then I think apart from having good ideas and writing a good piece of software and a method, then it's back to that point of benchmarking and doing really rigorous comparisons with what already exists to show that there is a need, and then that your particular method really does do a good job of solving the problem. And that's kind of one of my disappointments with some of those, especially those differential expression methods developed for single-cell RNA-seq data, is that they don't actually solve the problem they were set out to solve, and that kind of makes me sad. So don't do that.
You really need to be rigorous.
Yeah.
Thank you.
Thanks, Alice.